food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Robert Lustig, welcome back to the podcast.
Thanks very much for having me, Nick. Happy New Year.
Happy New Year. Yeah, one of my favorite past guests, one of the best episodes from maybe it was 2 years ago almost now. But there's a lot more that I can discuss with you. Some of it will be related to, I think, what we talked about the first time. I recommend to people go back and listen to that episode. And some of it will be brand new. I want to start off by talking about some basics of cellular metabolism. And we're going to go over some stuff in a fair amount of detail, but you've laid out in a review paper that I read some stuff in a really nice fashion in terms of talking about how some of the key regulatory pathways in cells work and how they can line up or be synchronized or desynchronized with each other to determine how much a cell is growing versus how much it's burning things for energy, how much it's sort of healthy and things are working in one of those two fashions the way they should be, and how these things can become dysregulated. But I want to start out pretty basic here. What is the difference between growth and burning modes that cells have, and how does that work at a high level?
All right. So, very good question. Think of it this way. We all start as a zygote, a single cell. Everyone, single cell. We end up an adult, 10 trillion cells. Now, that single cell had to divide. So now you have 2 cells. And then those 2 cells had to divide. Now you got 4 cells. And then those 4 cells had to divide. Now you got 8 cells. And on and on and on and on until you end up with an adult. Okay. 10 trillion cells. How many doublings did that zygote have to undergo to get to be that adult? So, simple exponential equation. 2 to the what equals 10 trillion? That's the question. So what's the number, Nick?
We can do the math. What is the answer we get?
Okay. 41.
41.
People are blown away by the idea that 41 doublings gets you to 10 trillion.
Yeah, the typical person might intuitively think it's going to be some extraordinarily large number.
Right. It's not. It's 41. Okay. Now, 2 to the 41 equals 10 trillion. Now, of those 41 doublings, some of them are going to occur in utero before birth, and some of them are going to occur postnatally after birth. So, how many of those 41 occur prenatally, and how many of them occur postnatally? So I need two numbers that add up to 41. What do you think?
I'm going to guess more prenatally than postnatally.
Yeah, that's a good guess. Like how many?
Solid majority.
36 and 5.
36 and 5. Okay.
36 and 5. Vast majority occur prenatally. Okay. So I can prove that too, by the way. Okay. Typical baby weighs 7 pounds at birth. Okay. First doubling, 14 pounds. Second doubling, 28 pounds. Third doubling, 56 pounds. Fourth doubling, 112 pounds. Fifth doubling, 224 pounds. Might be a patient with metabolic syndrome.
Right. But okay. Yeah. That is roughly.
Yeah. 5 doublings. 5 doublings after. So 36 before and 5 after. Now, is it possible that you might see a baby who, instead of having 36 doublings before birth, only had 35? What would that baby look like? Be small.
Yeah.
Right? Be half the size, right? What we call small for gestational age.
Mm.
Now, is it possible that that 36th doubling would occur after birth, so that instead of it's 36 and 5, it's 35 and 6? Yeah. In fact, 7/8 of the babies who are born SGA end up recouping that last doubling. They just happen to do it after birth instead of before. And so they end up normal size. Now, turns out 1/8 of them don't. They never get their last doubling back. So they only got 40 doublings, 35 and 5. So what do they look like when they're an adult? They're short, they're small. It turns out those are the patients who actually respond to growth hormone. And we give growth hormone to patients who are SGA, like, for instance, Russell-Silver syndrome, et cetera. And this is my job as a pediatric endocrinologist. And we can coax that last doubling out of them and increase their growth rate so that they end up as tall as they would have been otherwise. That's kind of cool when you think about it, isn't it?
Yeah.
All right. So, but what this is telling us is that there's 36 doublings before you're even born. There's a lot of growth going on before you're born. And in fact, the growth that we think is so important after birth is actually slowing down. Right? Now, that growth that's going on before birth is occurring in a uterus and a placenta. And what's the oxygen content of the blood that's feeding that fetus? It's really low. It's really low. Okay. In fact, the partial pressure of oxygen in the air that we're breathing right now is at 100 millimeters of mercury. The partial pressure of oxygen in a cancer, okay, we always say that, cancers don't need oxygen, the Warburg effect, which we're going to get to in a minute. Okay. The partial pressure of oxygen in a cancer is only like 54. The partial pressure of oxygen at the top of Mount Everest is 51. And the partial pressure of oxygen in the placenta, which is feeding the baby, is between 6 and 31. So a famous paper in 1954 actually, called this issue out and called the paper Mount Everest in Utero. So, in fact, growth does not need oxygen.
Yeah, the growth rate is higher in the more hypoxic environment.
Exactly. Whereas in the high oxygen environment, you see more burning. And the reason is because mitochondria need oxygen in order to be able to complete the Krebs cycle, in order to be able to generate the ATP needed for burning. So what this means is that there's a switch that's going on inside each cell to determine at any given moment, is this cell going to grow or is this cell going to burn? And you can't do both at the same time. So the question is, what's the switch?
And why can't you do both at the same time?
Well, it's kind of like you got a cord of wood out in your backyard. You can do one of two things with it. You can make furniture, or you can use it for firewood. But you can't do both.
Yeah, with any given piece of wood, it's got to be used for one or the other.
One or the other. And that's kind of the way it works for cells. In fact, the burning phase is actually not set up for being able to grow. And the growth phase is not really set up for being able to burn. And the reason for that is because in the growth phase, you're actually using glucose instead of for ATP, you're using it for precursors for growth. So glucose goes to lipids through de novo lipogenesis. Glucose goes to amino acids through the pentose phosphate shunt. Glucose goes through to nucleic acids through the Randall cycle. Okay, but the point is that you need the glucose intermediates like pyruvate and dihydroxyacetone phosphate, et cetera, in order to be able to create those things that are necessary to increase the number of cells.
Right. So the zygote's dividing. You need to make more cells. That means more lipid membranes. That means more proteins. You need to all of the Lego blocks that go into building these things, and you make them from glucose. And if you do things with glucose like that, then obviously you're not going to be using it to make ATP.
Exactly right. Exactly right. But you still need ATP because you need energy, right? I mean, that's the currency of the cell in terms of energy need. So it's not that the ATP is not needed. It is. Mm-hmm. The question is, if all of the glucose is going to growth, where's the ATP coming from? And that's the Warburg effect. That's, what Otto Warburg won the Nobel Prize in 1931 for realizing. Cancer cells do not need oxygen. And the reason is because they're growing. They're not burning, they're growing. But they still need ATP. So how do they do that? Well, as you remember from biochemistry, all of you, audience members out there who hopefully, took 10th grade biology, there are two steps in metabolism. The first step, you take glucose down to a molecule called pyruvate, and you do that in a process called glycolysis, anaerobic glycolysis.
No oxygen.
No oxygen. You don't need oxygen for any of those reactions. And that will take you all the way to pyruvate. And in the process of turning glucose into pyruvate, you will release 2 ATP for each molecule of glucose. Now, 2 ATP is not a lot. Now, from pyruvate, you then enter the mitochondria. The pyruvate gets converted to a compound called acetyl-CoA. The acetyl-CoA then participates in this Krebs cycle, tricarboxylic acid cycle, the cycle of, true energy production. And you get 36 ATP out of that.
Way more.
So you get way more ATP out of mitochondria than you do out of glycolysis. That is absolutely true. But you still get those two. Now, those two are not enough to run all the functions that a cancer cell needs. But what if, instead of importing a little bit of glucose, what if you imported a whole boatload of glucose?
Yeah. You can't convert as much of each glucose molecule into as much ATP, but you could get around that problem by just importing more glucose.
Exactly. And there's an enzyme in the cell that does that. And that enzyme is called PI3 kinase, phosphatidylinositol 3 kinase. And this is the enzyme that Lou Cantley, who was at Harvard now, of course, for many years now, Cornell, has demonstrated is one of the primary movers in cancer. And basically, all cancer cells ratchet up their PI3 kinase because PI3 kinase is the spigot that lets more glucose into the cell so that you can basically not burn, but ferment through glycolysis in order to be able to generate the ATP you need while still having precursor substrates that can be used for growth. And that's what cancer cells do.
Yeah. So you get growth and you get more energy at the same, you get around this trade-off that would otherwise happen in the cell, the growth versus the burning, by importing more glucose. And then from there, you can build as many Lego blocks as you need to grow, but also have enough energy to do it.
Exactly. And that's what cancer cells do. And that's why they're kind of cool. Well, it turns out that's what fetal cells do too. Cancer cells and fetal cells are almost identical. And so it turns out that there are three enzymes that basically control whether the cell is going to be in growing mode or whether the cell is going to be in burning mode. And those three enzymes are PI3 kinase, like I just talked about. That's the spigot that lets the extra glucose in. The second enzyme is AMP kinase, adenosine monophosphate kinase. Okay. And that is the fuel gauge on the cell. That basically tells the cell whether your energy replete or energy deplete. And if your energy deplete, then you want to basically build more mitochondria in order to generate more energy. So that is the primary signal for mitochondrial biogenesis. And then the third enzyme is called mTOR, mammalian target of rapamycin, which I'm sure you have talked about on your show many, many times because it's really cool. It's where rapamycin works, which, basically alters the immune system and basically people think is the fountain of youth and what have you. And the reason is because rapamycin stops growth. And when you stop growth, okay, less chance for cancer. Unfortunately, when you stop growth, you also stop the immune system, in which case more chance for infection and death. And so this is, of course, the yin yang of why rapamycin is not routinely used in longevity clinics. At least reputable longevity clinics.
So basically, we've got three different pathways in any given cell, and how active or inactive they are relative to each other is going to control the extent to which the cell is going to be more in the growth mode and build new stuff mode, or the make energy in the form of ATP mode. Right. And the oxygen content of the environment is really important for regulating the balance of those three pathways.
Right, because the oxygen is needed for the mitochondria. So the mitochondria can't work without oxygen. That is completely true. Okay, so it turns out oxygen is one of the organizing principles. But when you don't have oxygen, it's not like your cells die. They just go into fermentation mode. They go into growth mode.
Yeah, because that's just what they have to work with and what they're limited by.
That's exactly right. So that's kind of cool when you think about it. All right? But most of that growth is happening before you're even born. And, people sort of miss that because you go from a single cell to 7 pounds. Okay. Well, that single cell to 7 pounds, I don't know what the infinite amount of increase in volume or in mass that is, but it's way more than going from 7 pounds to 224 pounds. All right. So those are the principles by which growth versus burning occur. Now, you mentioned this question of synchrony. You have these three enzymes, right? PI3 kinase, AMP kinase, mTOR. They are all kinases.
And so a kinase is just a particular type of enzyme that does what exactly?
Adds a phosphate. Kinase, K, phosphate. Okay? So it adds a phosphate. So what it does is it changes the phosphorylation status of other enzymes. That's what it does. Okay? And what it's doing is changing how energy is being utilized and what's happening to it inside the cell. And what we now know is that because there are three enzymes and there are two valences on off for each of those, that means there are 8 permutations. So one permutation is the growth permutation. That would be, high PI3 kinase, low AMP kinase, high mTOR. Okay. Then another one would be burning. That would be the opposite. That would be low PI3 kinase, high AMP kinase, low mTOR. Okay. Well, now you got six more. You got six more permutations where they are now dysynchronized. They're not driving growth and they're not driving burning. They're not hooked up with each other the way growth or burning would be.
Yeah. So basically, there's two basic states each of the three pathways can get into. So that's 2 to 3, that's 8. And if they're, you sort of want them to be either in growth mode, which is going to be one constellation of these pathways being on, and then you want them to be in burn mode. But then there's a bunch of ways left over that they can be dysregulated or out of sync with each other.
Out of sync with each other. And it turns out all of those lead to metabolic disease of various sorts. Could be cell senescence, could be inflammation, it could be metabolic syndrome, could be obesity. Okay. Bottom line is those are where these diseases of modernity come from, is the inability of these three enzymes to link up and do their job in concert with each other.
Yeah, in concert. They have to be in a certain balance relative to each other. And just so we don't lose track of it, and so people remember with some repetition here, what are these three cellular pathways again? And what's sort of the analogy? You've got PI3 kinase, you've got AMP kinase, and you've got mTOR. And what
That's right. So PI3 kinase lets the glucose in.
How much substrate is coming into the cell?
Right. AMP kinase is building new mitochondria to make more energy.
How much ATP can I make?
How much can I make to be in burning mode? And finally, mTOR is whether or not the cell is ready to divide.
Yep. Build stuff, grow, anabolic pathway.
Right. Okay. So when your PI3 kinase is high, you're importing a lot of glucose. When your AMP kinase is low, that means you're not funneling into mitochondria. So you have the precursor substrate of pyruvate to be able to go into, for instance, membranes, or you have the precursors like DHAP, which could go into the Randall cycle or into the pentose phosphate shunt to make other things that the cell needs. And then finally, mTOR, which basically says, okay, we have everything we need here to build a new cell, divide. Okay, and when those three enzymes are hooked up and in concert with each other and synchronized and in growth mode, you grow. And when they're in the opposite, then you burn. And the point is, growth is necessary at certain points in your life, and burning is necessary at other points in your life. And every cell in your body at some point in time had to grow, and it also had to burn. And it is these relationship of these substrates to each other that determine that. Kind of cool.
So you've got three pathways. They can be aligned or disaligned in a few different ways, a few different patterns.
Right.
So there's one way they can be aligned that puts you in growth mode. Let's call it healthy growth mode. They can be aligned in a way that puts you in healthy burning mode. And then they can be misaligned in a number of ways that are going to lead to various kinds of dysfunction. And the oxygen content and availability of the environment is an important signal for which mode you're in and potentially whether or not things become disrupted or misaligned.
Exactly right.
And then one way to think about the oxygen is the developmental angle. That's what we were talking about. The fetus is relatively hypoxic. That favors growth mode over energy mode. And then you're born, the oxygen environment goes up, and then you flip more from growth mode into burning mode. Would another lens to maybe think about this that people can attach their minds to be something like exercise and resistance training?
Yeah, absolutely. So if you go to the gym, okay, one of the things that they now do for older people is they do something called blood flow restriction bands. Okay. They put bands on their arms and their legs very specifically to increase the ischemia, increase the hypoxia of the extremities, because what that does is that then builds muscle. So this whole issue about protein and sarcopenia and its relation to mortality, has made its way into the gym. And so we have this concept of blood flow restriction increasing muscle mass and muscle size, specifically to try to solve the sarcopenia problem. And the reason that works is because you're depriving those muscles of oxygen. So instead of burning that glucose, they're going to use it to grow instead.
Would that be like why when you talk to someone like a weightlifter and they're talking about growing and hypertrophy of muscles, they always talk about going to failure and how it's those last 1 or 2 reps that you can't quite do that are going to really stimulate the growth because
exactly.
In the constellation of these enzyme pathways, that would be, you're basically putting your cells into a high mTOR because you're also probably eating a lot, high mTOR and then low AMP kinase state. So they're saying import the glucose and use it for building stuff.
That's right. Exactly right. So, you can use it, this concept to basically sculpt your body, and potentially even, improve longevity. So, this is an exciting potential, pathway for improving health and well-being and, of course, muscle mass. And, we'll talk in a little bit about one of the trade-offs of that. And that is that you're not making stuff in your mitochondria. Okay. And one of the things you're not making in your mitochondria is something called reactive oxygen species. And we're going to spend a whole lot of time talking about that, I guess.
When we think about the earlier stages of development, so in utero, from single zygote in this relatively hypoxic state, this growth state that the developing fetus is in, what are the origins of metabolic dysfunction in the developing fetus, and how much of it has to do with the environment the fetus is in while it's inside the mother versus the metabolic state that the mother was already in by the time she got pregnant? Can we talk a little bit about that?
I'm not sure there's a difference between the two. I think they're the same. I think the metabolic state of the mother is the environment that the fetus finds itself in. I think that's exactly right.
I guess what I was thinking of there is, let's imagine the following two situations. A woman gets pregnant. Maybe she's not the most healthy, but maybe she's not in horrible shape, whatever that means. She could take one of two paths. She could say like, well, I'm just going to eat whatever I want while I'm pregnant and do it, like continue in her, somewhat unhealthy state during the pregnancy. Or on day one, she could say, I'm going to be really healthy because, I want the baby to grow well. And so she changes her state. So let's say this was an obese woman who continued doing what she was doing before she was pregnant during the pregnancy, or an obese woman that was already obese, but then at the time of pregnancy, she starts changing her lifestyle. Mm-hmm. Is the fact that she was already obese or metabolically dysfunctional in various ways at the time of conception going to have some effect, or is it all about what she does after that?
That's a very good question. My colleagues here at UCSF are very interested in that because we think that that has a lot to do with the problems of assisted reproductive technologies, ART. Okay, so you're familiar with intrauterine insemination and in vitro fertilization, ICSI, intracytoplasmic sperm injection, et cetera. There are a whole bunch of different ways that reproductive endocrinologists can help people get and hopefully stay pregnant. Right? Well, turns out that if you look at the data, and we have, the congenital malformation rate in a set of normal pregnancies is about 3%. Okay, now different things within that congenital malformation rate of 3%. But if you look at babies born from assisted reproductive technologies, that number is closer to 9%. So it's almost triple. Okay, and it's a big problem. Big issue. And we're trying to figure out why and how to solve that. Is it, you're incubating the embryos in a broth of, growth factors, and maybe you don't have the right ones or right amounts, et cetera. That's one possibility. It's also a possibility you're not making enough ATP because you don't have the oxygen concentration just right, et cetera, et cetera. There's a whole host of potential things that could be going wrong that would lead to this tripling in in congenital malformation rate, and we're trying to figure out what those are. And one possibility is that the mother, because she was metabolically unhealthy, and that's why she needs assisted reproductive technologies, carried that over in terms of the egg in the first place. Or the other possibility is that it's the broth that the baby is, that the embryo is being bathed in that leads to that. And that's really important because it kind of it sort of, addresses the question of, can mothers fix their metabolic parameters so as to improve their infants' chances? Now, we know for a fact that if mothers gain a lot of weight during the pregnancy, their risk for gestational diabetes goes up and the risk for congenital malformations goes up. And those babies are at risk for obesity and metabolic syndrome in later life. We know that. We also know that smoking, which causes reduction in fetal growth, also leads to problems later in life as well. So it can go either way. You can be too big or you can be too small. In addition, what the mother eats is extremely important as well. So glucose control, during the pregnancy is what we're finding that's extremely important as well. But the question is, that you're asking is, what about the glucose concentration right at the time of fertilization? Is it like too late already?
Yeah. Or even before that, is something about the oocyte that was unfertilized, right, different than that could have been if she was in a different metabolic state?
Exactly. And we don't know the answer to that. We just do not know the answer to that yet. Having said that, my colleague, Dr. Bruce Blumberg at UC Irvine, who is a wonderful guy, you can always find him because he is the one who goes to every meeting in a Hawaiian shirt. I love Bruce.
Yeah, no, he's been on, and I highly recommend people check out that episode because it's very well done stuff.
He's super. And what he's been doing is he's been looking at compounds in the environment that lead to changes in growth during, in utero before birth that ultimately get carried forward from one generation to the other. So it may not even be what the mother did during her pregnancy, may be what her grandmother did 2 generations earlier, or even what her great-great-grandmother did 4 generations earlier, that is now having an impact on her fetus that she didn't even know anything about and couldn't control even if she wanted to. So an example of this is a compound that Bruce has been studying for years called tributyltin, TBT. And what this does is it causes major metabolic havoc in the offspring. And it causes diabetes, and it causes fatty liver and all sorts of things. It's bad stuff. And the problem is it's everywhere because it's what you paint on the bottom of your boats to keep barnacles from attaching to the hull. So it's throughout the entire water supply of the United States.
People drink it, basically.
Yeah, people drink TBT like every friggin' day. Okay, you cannot get away from it. It is there. And what he's shown is that this compound called tributyltin affects an enzyme, the expression of an enzyme in the liver. And that enzyme is called insulin degrading enzyme, IDE. And it basically changes the epigenetic folds of the chromosome where that enzyme is. And basically prevents it from being expressed. And it stays that way.
So if it's insulin degrading enzyme, then you're going to get more insulin, and more insulin is going to mean more adiposity.
More obesity. So this is an obesogen. This is a compound that drives obesity that has nothing to do with nutrients. It has nothing to do with diet.
Nothing to do with calories. Nothing to do with what you did. It could be
or exercise
generation.
Right. And it's not, it has nothing to do with anything you did. It's what your grandmother or your great-great-grandmother did.
We're talking about a chemical that induces a gene expression change that essentially makes your cells stuck in an exaggerated make fat mode.
Exactly. So we call this an environmental obesogen. Okay, a compound that drives adiposity unrelated to its caloric equivalent. That's an obesogen. Okay, so you can't do anything about it. It's baked into the cake. And it may be one of the reasons for the obesity epidemic, sort of having gone hog wild over these past 40, 50 years, because we are now exposed to all of these obesogens. And by the way, the obesogens predate, the obesity epidemic by about 10 years, which you would expect to see if something was causational. So this is very, very worrisome, very important, but it also, has direct implications for how we can prevent all of these things from happening once we understand what the actual cause is.
And I definitely want to talk more about chemical obesogens, and I think we will do that in different ways. I want to talk a little bit more about development, and in particular, how the mother's nutrition affects development in terms of calories and macronutrients that people might be familiar with, things like fructose, things like various fatty acids. So can you start to talk about this? So in terms of caloric obesogens, stuff that we consider to be food, macronutrients, when a woman is pregnant, what are some of the more salient ways that modern diets are exposing the developing fetus to things that are going to favor metabolic dysfunction and the desynchronization of those three enzyme pathways we talked about?
Absolutely, sure. So obviously people eat and some of that food goes to the fetus, right? Through the placenta, as individual nutrients, whether it's amino acids, whether it's fatty acids, whether it's, nucleic acids, whether it's fructose, we're going to get to in a minute. Okay. And then the cell basically takes those in, the fetal cell takes those in and uses them as building blocks for generating new cells in the absence of oxygen, like we just talked about. All right. So what determines whether a cell's going to burn? What's going to determine if a cell is going to grow? And then finally, remember those 6 other parameters, the 6 other paradigms, where they're not lined up? What happens there? Instead of growing or burning, you're not. What are you doing? You're storing. So what causes a fetal cell to store? How can a baby be born with a fatty liver?
Right.
How's that? That's the easiest way to, sort of bring this home. Okay. The fact of the matter is we now have an epidemic of neonatal obesity.
Yeah. So not just childhood obesity, but neonatal obesity. Yeah.
Okay. 4 separate studies, secular trends in birth weight in 4 countries, South Africa, Israel, Russia, United States, over the last 25 years, there's been an increase in birth weight of 200 grams over 25 years. Now, what are those 200 grams? Well, if you stick those babies, those newborns, in a scanner, in a DEXA scanner, turns out it's all fat. Mm-hmm. These babies are laying down fat. And they haven't, had a drop of mother's milk yet. We have neonatal obesity. We have neonatal fatty liver. How do you get fat in your liver when you're a newborn? So this is the question: is how does this occur? So clearly, you're not growing, and you're not burning. So you must be in one of the other 6 paradigms. And it turns out that yes, indeed, in order for that to occur, your mitochondria are clearly not able to process the energy that's trying to be driven through it. Your mitochondria are diverting. They are diverting away from ATP and towards storage. And the question is, how come? Well, mitochondria are not doing their job. We have a pandemic of mitochondrial dysfunction across the board. Yeah. And it's been going on for a very, very long time. Now, how do I know this? Paper came out in 2020. First author is Protsiv, Protsiv. And what looked at was the basal body temperature of the United States for the last 150 years. So they all went all the way back to Civil War records to find out what is a normal body temperature. And it turns out that in the late 1800s, a normal body temperature was 98.6 degrees Fahrenheit or 37 degrees centigrade, just like what you learn in, elementary school today. Except it's not true anymore. Because over the last 150 years, and it doesn't matter if it's women or men or black or white or old or young or anything, okay, every demographic that they looked at, okay, that 98.6 degrees basal body temperature is now 97.0 degrees. And it's been going down consistently over time over the last 150 years. Mm-hmm. We burn, and your body temperature is basically what your mitochondria can't capture as ATP.
Right. So, literally, they're burning. It's going to give off heat. That's why mitochondrial function and capacity is going to be related to what your overall body temperature is.
That's right. That's right. So, bottom line, this is proof positive that mitochondrial function is not what it used to be. We have some modicum of mitochondrial dysfunction that's going on all over the country, really all over the world. So what is causing our mitochondria to not work so well? Okay, think about it. If you process a bunch of glucose and you get a bunch of ATP, okay, now what if your mitochondria work 1% less well? You're processing the same amount of glucose, but if they're working 1% less well, what's going to happen to the 1%? It's going to go to fat.
Left over, to go to fat.
Okay. What if you are, burning 2% less well? You make more fat. And then that fat has to be basically packaged and exported out. And so that becomes triglyceride. And your triglyceride basically goes up, and that goes into, muscle cells. And now you have muscle fat and muscle insulin resistance. And you have liver insulin resistance. Ultimately, you get brain insulin resistance. And look, you have the entire panoply of metabolic syndrome. All because your mitochondria are not doing their job the way they should.
Mm-hmm. And so obviously the next question is, what exactly is breaking within the mitochondria, and why did it break? What was causing it?
So there we are. This to me is the single most important question in health, both systemic health, mental health, and societal health, that we've ever had to deal with in the entire history of medicine. This is the question. What's going wrong with our mitochondria, and what the hell do we do about it?
Mm-hmm. One way to start to think about this would be to ask a question such as, what is different about the composition of the nutrients going into the mother and ultimately into the fetal environment, or going into the mother and becoming her milk, or, compared to, mother's milk, natural mother's milk with modern nutrient intake versus formula and these things. Is there anything, what are the most salient things that jump out in terms of how the nutrients that are getting into the developing baby, both in utero and shortly after it's born and as it's growing up? What are the most salient differences in the inputs, the nutritional inputs today versus back in the day?
So I could sum this up in one word and drop the mic and walk away, but it's much more nuanced than that. And we'll get into the nuance in a minute. The word is fructose. Why?
That's one word.
That's why fructose? Why, my hobby horse, the thing I've been beating for the last, 18 years now. Why fructose? And the answer is because that's the thing that's gone up 250% in everybody, and 2,500% in some. Okay, somewhere between 250 and 2,500% in terms of diet. Okay, our fat content has not changed that much.
Total fat content.
Total fat content.
Total fat content.
Yeah, well, and we'll talk about individual fats in a minute because obviously mitochondria like certain fats and not others. We'll talk about that in a minute. Okay, but fructose is a dose-dependent mitochondrial toxin. And it's the thing that's been going up 250 to 2,500%.
And I want to pause here and give you a chance to unpack that because we've talked about it before, but also there's a lot of chatter online and some people take issue with this. So can you just define precisely dose-dependent mitochondrial toxin and emphasize very clearly, if you can, the dose-dependent part of it?
Yeah. So it's dose-dependent like alcohol. Okay. Alcohol is dose-dependent as well. All right. Bottom line. Fructose has calories, 4.2 calories per gram. Yeah, I know. They do. That's only if you burn them to ATP. What if you don't burn them to ATP? What if actually fructose stopped burning? Well, in fact, it actually does that. How does it do that? Well, a metabolite of fructose, which I mentioned earlier, called dihydroxyacetone phosphate, DHAP, in the liver, okay, where it's being metabolized, so fructose goes to fructose 1-phosphate and then ultimately gets cleaved to glyceraldehyde 3-phosphate one direction and dihydroxyacetone phosphate the other direction. Okay, that dihydroxyacetone phosphate has the opportunity to become a compound called methylglyoxal, MgO, methylglyoxal. And methylglyoxal has an Al at the end, which means it's an aldehyde. Okay, so everything that's an aldehyde has an Al at the end. Methylglyoxal has an aldehyde. That aldehyde, and it's now a 3-carbon fragment, methylglyoxal, has an aldehyde, and it fits right in the active site of the gamma subunit of AMP kinase.
So that's one of the 3 major pathways we talked about, which regulates whether or not a cell is going to be in growth versus burn mode.
Exactly right. And that AMP kinase determines whether or not you're going to build new mitochondria. Well, this aldehyde, this methylglyoxal, insinuates itself right into that gamma subunit where the AMP should be in order to generate a, increase in activity and therefore an increase in number of mitochondria. Right.
So if you mess with that, would do by having too much fructose around, you won't make as many mitochondria and therefore you won't be able to handle as much glucose and therefore it'll be left over to make fat.
Number one. And number two, that enzyme that it bound into the gamma subunit, that aldehyde will react with one of the 3 arginines in that active site and will form a covalent bond. Okay, just like the Amadori rearrangement. Basically, now that methylglyoxal can't come out of that active site.
You just broke the little machine.
You broke the machine, right? It's irreversible inhibition. It's called in the in biochemistry, irreversible inhibition. Okay, you've killed that enzyme, and then you have to make a new enzyme.
Right.
Except you're not going to make a new enzyme because you turned the friggin' thing off.
Yeah, you're having an energy production problem. And there's always a trade-off. There's only so much energy to do so many things. If you've got to make a new enzyme because you broke it, well, you need to be able to make it. You might not be able to do that. But even if you could, that energy would have gone to something else. And there's a lot it could have gone to in a developing fetus.
Exactly. So what's going to happen now to that, to the energy that was being poured down that glycolytic pathway to hopefully get the pyruvate in order to be able to enter the mitochondria, which is now friggin' broken? Well, there's only one thing that can happen. It's going to get turned into fat. It's going to be diverted. It's going to be stored. And so you now have a broken mitochondrion. You have the inability to create a new mitochondrion. And you have the diversionary pathway taking all of the excess substrate and marching it off to storage. And that is why fructose is a dose-dependent mitochondrial toxin. And by the way, so is alcohol. because
Right.
alcohol does the same thing.
Right. Some of these things will only happen if there's enough fructose around. It won't
Yeah,
happen
yeah,
if there's a smaller amount around.
yeah, absolutely. Of course, it's dose dependent. Right. So, but the problem is that our fructose consumption has gone up 250 to 2,500% because it's in everything. And we like it too because it's, sweet and it's also addictive.
Yeah. Yeah. One of the things I've always found confusing is um, psychologically confusing about people. I mean, we always talk about things like high fructose corn syrup, and people will say, well, it's 55-45. Sucrose is 50-50. It's only this
Well, it's
5%
true.
difference,
Yeah,
but it's in everything. And you're
but it's
eating
in
it every
everything.
day. So times
That's right.
X number of doses per day times
Yeah,
X number of days per year for a lifetime. And now
yeah,
you've got a big number.
yeah. I completely agree. And of course, it's not really 55-45. Michael Gorin and Elsie Ventura years ago went around Los Angeles. So they're both the children's of LA. Went around Los Angeles and actually sampled the soft drinks coming out of the 7-Eleven dispensers and sent them off for analysis. And it turns out that a lot of those were 65-35,
Hmm.
65 fructose. And the reason is because you use 90% fructose, high fructose corn syrup in those dispensers. And so, if you have those fine tuned the wrong way, you're actually dumping more sweetener into there. And so that's actually making things even worse. Now, it's not happening in a can of Coke, but that could be happening in the Coca-Cola out of a 7-Eleven machine.
Hmm. Interesting. Interesting. Okay. So fructose has dose dependent effects. And under certain conditions, it can break some of the key enzymes that we've been talking about that
Right.
regulate what the cells are doing and whether or not they're in burn mode versus growth mode. The mitochondria are super important here because this is where oxygen goes. We breathe oxygen so it can get into the mitochondria to
Yep.
do mitochondria stuff. Of the three enzyme pathways we talked about, just to give people a sense, are those enzymes literally inside the mitochondria? Are they elsewhere? Are they breaking sort of in the mitochondria, near the mitochondria?
So no, they're outside the mitochondria. None of them are in the mitochondria per se themselves. There's other stuff going on in the mitochondria that can also be broken.
Mm-hmm.
But fructose that's how fructose does its problem. Okay. And so the more sugar in the diet, the more mitochondrial dysfunction, the more energy diversion to storage, the more fat, the more deposition of fat, and the more insulin resistance, which then drives all these chronic metabolic diseases.
Mm-hmm.
So fructose is kind of the big kahuna on the block in terms of this process, this phenomenon, this, debacle
Mm-hmm.
that we see.
Yeah.
And
And so the
it's the
thing,
one we could fix tomorrow if we had the political will to do it.
yeah.
That's why I focus on it.
And so the process that we just talked about, where fructose dose dependently breaks some of these key enzymes, it's breaking these enzymes outside the mitochondria, but these are pathways within the cell that are involved in telling the cell to make more mitochondria or not,
Right.
depending on the environmental demands placed on the cell. So if you
Right.
break that signal outside the mitochondria, you are preventing the cell from making more mitochondria if it needs to, and it often needs to in today's world. And so that leads to problems.
Right.
What and how do the mitochondria actually break themselves?
Okay. So what's in the mitochondria that
Yeah.
can break? That's what you're asking. And the answer there is fats. So think of it this way. Mitochondria, as you know, are refurbished bacteria. Okay. They have their own DNA, mitochondrial DNA. And of course, that DNA comes from the mother's mitochondria, not from the father's. So obviously, if the mother's mitochondria are a problem, then the baby's mitochondria will also be a problem. That's also true. What are the mitochondria made of? Well, they're made of fats. They're made of of unsaturated fats. Now, different people eat different fats, but the fats that the mitochondria are made of are all essential fatty acids. You have to eat them.
So you mean,
don't make them.
you mean there's proportionally more unsaturated fatty acids in the mitochondrial membrane than in the cell membrane?
Correct. That is correct. Okay. Those, and it turns out that the number of double bonds in those unsaturations have a lot to do with mitochondrial efficiency. Okay. This is the mitochondrial fats as a conglomerate are known as cardiolipin. Okay. And because they are fats of the heart, if you will, cardiolipin, fats of the heart. Okay. That's what the mitochondria are, is the fat of the heart, because there's no fat in the heart other than in the mitochondria, because the heart's working really hard. It's all muscle. Okay. But the fat in the heart is cardiolipin. And the cardiolipin, okay, the energy transfer to ATP is dependent on the number of double bonds. There's a certain sweet spot for double bonds, 2. So if a fatty acid has 2 double bonds and they're cis, not trans, okay, so trans fats, total disaster. But if they're cis, okay, you get mitochondria that do their job and do them well. If they're only 1 double bond, they work less well. If they're 3 double bonds, they work less well. 2 double bonds is like the perfect one. Okay. You know what those are called? Those are called omega-3s. That's what omega-3s are. So omega-3s are the source of cardiolipin, and cardiolipin is the source of energy efficiency inside mitochondria for being able to turn energy, chemical energy into ATP for the cell to be able to use.
So the fat that the mitochondria are made out of is important. In particular, the number of double bonds in these fatty acids is important for mitochondrial function and efficiency.
Correct.
In simplified terms, you just emphasize 2. So if you have 3, that can be bad. If they have a trans configuration, that can be bad. If they have only 1, that can be bad or less efficient. But there are many ways for fatty acids to have 2 double bonds. And you mentioned omega-3s as good.
And
what if there's another what if there's another pattern of 2 double bond fatty acids that are, say, very prevalent in the modern industrial diet that start to get in there?
So what are we talking about? Seed oils.
Yeah.
Yeah. So the seed oils are interesting. And of course, everybody and his brother, are clamoring about seed oils right now. And you've had several guests on, talking about seed oils in the past, and I've listened to those as well. I have a bone to pick with it, a little bit, anyway. So first of all, seed oils are a source of omega-6 fatty acids, not omega-3 fatty acids. Omega-6s are important for the immune system. They are the precursor to arachidonic acid. And arachidonic acid is the precursor to thromboxanes, leukotrienes, and
prostaglandins.
Huh?
Prostaglandins.
Prostaglandins. Thank you. Okay. Thank you. So you need leukotrienes and thromboxanes and prostaglandins in order to be able to generate an immune response. If you can't generate that immune response, you will be eaten by the maggots. Okay. So you got to have omega-6s. So I'm not against omega-6s per se.
Yeah. You have to have enough of them.
You have to have enough of them. And the problem, of course, is we have too much of them. And I agree with that. We have way too much of them, 20-1 instead of like 1-1 or to 3-1, which is what, our ancestors used to consume, And so you can set yourself up to be in a pro-inflammatory state with omega-6s. Omega-6s are important for the immune system, and I'm not saying they're not. Okay. But too many of them basically set you up to be in a potentially pro-immune or pro-inflammatory state, which is not good. There's another problem with omega-6s, which is not discussed. And this, I think, is where your guests have gotten it wrong. If you look at what happens to omega-6s, first of all, omega-6s are cheap because seed oils are cheap. Soybean oil is cheap. And so virtually every ultra-processed food on the planet uses omega-6s as its base because they're cheap. Now, omega-3s burn easy. If you heat them, number one, they go rancid, they flip and become trans fats. And we know that. And it doesn't taste good. I mean, basically, try taking, algal oil, which is an omega-3, okay, and heat it, super high in a frying pan, okay, it'll burn really fast. And it's got a low smoking point to boot. Olive oil is a monounsaturated fatty acid. It also has a low smoking point, and it'll burn quickly also. Now, saturated fat doesn't burn, but saturated fat doesn't have any of these benefits either. And of course, it's animals, and people don't want animals in their Cheetos. So omega-6s are the overwhelming favorite of the food industry for ultra-processed food snacks because they can be heated to a high level. Here's the problem. If you look at the smoking point curve, and if you look at the trans-flipping curve of omega-6s, at 200 degrees centigrade, 400 degrees Fahrenheit, you get just as much flipping and you get just as much burning as you do from any of the others. In other words, you're not buying the benefit you thought you were. And you're basically ending up with a problem in terms of, cell dysfunction, mm-hmm. because those abnormal fatty acids that have been flipped will be ingested and will basically not be able to be processed and cut up into 2 carbon fragments, and will end up, causing problems as well.
Yeah, we'll end up breaking stuff in the cell.
Yes, exactly. So the omega-6 story is much more complicated than what the people who are, currently railing against it, discuss. So I'm not for omega-6s. I'm not against omega-6s per se. What I'm against is heating them. And that's what's going on in ultra-processed foods.
But can't they be heated inside the body as well?
No, not to the same extent.
Yeah, our bodies don't get to the boiling temperatures of a McDonald's fryer.
Exactly.
But at some rate, these things are going to be exposed to oxygen, and they can undergo lipid peroxidation. And we know that you can measure plenty of lipid peroxidation products that come only from omega-6 fatty acids.
Well, the question is, was it from omega-6s at room temperature, or was it omega-6s that have been preheated? That's my question. And the data on the preheating is very clear. So that's what I think is the big problem. So I am not for seed oils, but it's because of the fact that they are used in extruders like Cheetos, making Cheetos, as opposed to just being soybean oil in a salad dressing. So that I think is part of the mitochondrial issue as well. What else? What else can break mitochondria? Well, branched chain amino acids can break mitochondria too. Now, how does it do that? So branched chain amino acids are leucine, isoleucine, and valine. Now, they are 20% of muscle. They are what's in protein powder. They are what bodybuilders are, putting into their smoothies, when they go work out, in order to provide the amino acids to be able to build new muscle. And if you're a bodybuilder, that's exactly right. That's what you need. But what if you're not a bodybuilder? What if you're a mere mortal like me and you're consuming excess branched chain amino acids? What happens? Well, you're not building muscle. There's no place to store those amino acids. So what happens to the excess? What happens to the substrate that's not going to building muscle? Answer: goes to the liver. The liver deamidates it, takes the amino group off. Okay, so now instead of a branched chain amino acid, you have a branched chain organic acid. Okay, for instance, oxaloacetate, which then funnels into the Krebs cycle, overwhelms that Krebs cycle, so that it can't process, it can't run at a Vmax that's higher than it already is. And so what that's going to do, that's going to divert. And so you're going to end up with more fat being made. And so branched chain amino acid excess is associated with liver fat and insulin resistance and chronic metabolic disease as well. So, fructose, omega-6s, branched chain amino acids, these are all calorically laden substrates that lead to mitochondrial dysfunction. And they are all standard parts of the Western diet. That's
and again, kind of the point of a lot of this is, by whatever means, if you break the mitochondria and they can't as effectively transform glucose into ATP, then there will be more glucose left over, and that's going to basically put you in a state of laying down more fat.
Exactly. Well, it's also going to cause an increase in insulin release, which is going to cause energy diversion on its own. So basically, you've taken a well-oiled system and you've basically, introduced sand into the gears.
In terms of, when we think about nutrition during pregnancy and for the infant, so is it fair to say that basically we've pushed things in this direction as well when it comes to what the mother's eating and therefore the breast milk composition when it comes to infant formulas and stuff? How do things like the fatty acid and the sugar content of those things line up here? Is it in this direction of they've got more of all of the things that are sand in the gear?
Yeah, absolutely. So first of all, we used to think there was no fructose in breast milk. Wrong. If mom drinks a Coke, the fructose is in the breast milk. We used to think fructose didn't cross the placenta. Wrong. It crosses the placenta just fine. Why did we think it didn't cross the placenta? Because the placenta does not have the GLUT5 transporter. That's true, it does not. The GLUT5 transporter is specific for fructose. I agree, it does not have the GLUT5 transporter. It has the GLUT7, 9, and 11 transporters, which do both glucose and fructose equally well. And so there's plenty of fructose in the fetus. So, we thought we understood something and, we got it wrong. And so this is still a problem. So fructose is a big problem. Now, what about fats in the breast milk? Okay. In general, the mom is making the fats in the breast directly from precursors. Okay. And it's also doing the same thing with galactose. So it's turning glucose into galactose because galactose is necessary for brain growth of the baby, because the galactose is going to get turned into galactocerebrosides and ceramides in order to basically lay down myelin in order for the brain to myelinate and ultimately grow. In addition, galactose is necessary for the immune system to develop properly. If you don't have galactose, the immune system develops improperly. Example of that is galactosemia. So galactosemia, you can't turn galactose into galactose 1-phosphate. So you can't make the immune modulators that are necessary that galactose normally uses. And so those babies die by age 2 months of E. coli meningitis because they couldn't incorporate galactose into their brain. So galactose is super important, and the breast tissue makes the galactose. It also makes the fats in situ right there at the breast in order to be able to feed, the baby the appropriate fat composition of the milk. In addition, the breast also makes these carbohydrates known as human milk oligosaccharides, HMOs, which are 4 to 5 carbohydrate long little fibers, okay? Because they're not digested and absorbed. But what they do is they act like maternal antibiotics to keep the microbiome from going hog wild and and preventing pathogens from being able to set up shop in the baby's intestine. We know that because of this phenomenon that occurs in preemies called necrotizing enterocolitis, or NEC, which kills preemies, used to kill them right, left, and center. It was horrible, like 50%, death rate from necrotizing enterocolitis, something we pediatricians took care of routinely. But now we give the baby human milk oligosaccharides, and the percent of NEC has gone way down, and the percent of death has gone down even lower because these carbohydrate molecules, these strung together carbohydrate molecules, and there are like 200 different ones, are actually protecting the infant's gut. So there's a whole bunch of things the breast makes that are absolutely essential. But if the mother's not breastfeeding, then the baby's likely not getting those.
Got it. So infant formula could have things that are not supposed to be in there, wouldn't be in natural breast milk, or
like fructose,
or exist in a higher proportion. It can also be missing things that are in breast milk.
Yep.
So there's stuff that can be absent that should be present, and stuff that can be present too much or present at all that wouldn't otherwise be there.
All the above. That's why breast is best. Now, breast is not perfect. Recognize that breast milk does not have iron. Breast milk does not have vitamin D. Breast milk does not have fluoride. Not that fluoride is needed. That's another story. We can talk about fluoride till the cows come home. But breast milk is missing certain things that we, say are absolutely essential for babies, like iron and vitamin D.
How did babies survive in the past without supplemental iron
They
and vitamin
sort
D?
of didn't. There was a very high mortality rate
From the lack of iron and vitamin D in milk?
Yeah, sometimes, yes. Okay. The lack of iron, if the baby has a lack of iron, then that anemia leads to all sorts of problems with CNS development. This is shown by researchers at the University of Minnesota.
Yeah. But I mean, some of the babies survived in the past because we're all still here.
A few. But the infant mortality rate was enormous. Now, just so happens, infant mortality rate in developed countries has gone way down. I mean, our infant mortality rate used to be on the order of something about 50 per 1,000, and then it went down to 10 per 1,000. And now in developed countries around the world, it's more like 2 or 3 per 1,000. Ours is up at like 7 to 8 per 1,000. So even though we're a developed country, our infant mortality rate is much higher than all of the other OECD countries because we're not taking care of our pregnant mothers and fetuses and babies the way we should.
No, I understand that.
Part of it's our diet.
That's a great thing. Going from 50 to lower than 50 out of 1,000 is great, but 950 babies survived. I mean, were they all anemic and vitamin D deficient? And
No. I mean, a lot of people were out in the sun. They got plenty of vitamin D. They used to eat meat. they got their iron. But iron deficiency has always been a problem for mothers. That's just the nature of menstruation. You've got to be able to get the iron from somewhere.
Got it. Okay, so there might be things that are lacking in the breast milk that continue to be lacking in the modern environment unless we supplement for them, but may not have been lacking as much in past times in history because people were getting it by other means.
Indeed.
One thing I want to ask you about is the relationship between invisible microscopic stuff that we can't see with the naked eye as it relates to metabolic health and physical skeletal macroscopic stuff that we can see. When babies are born or throughout development, right, the body's physically being constructed. There's stuff happening at the cellular metabolic level. Obviously, we've touched on some of that. And then, the physical macroscopic features also development. The skeleton forms, the musculoskeletal system comes online. What is the relationship between the invisible stuff at the level of cellular metabolism and the macroscopic ways that development can go wrong? Does a macroscopic issue like a malformed skeleton have something to do ultimately with how well the metabolism is working at the cellular level?
Wow. Okay. So there are integrins, there are proteins that basically are working in a paracrine fashion, cell to cell communication that basically sort of lay down these patterns of growth that ultimately lead to our different organ systems that, I actually don't know that much about. And by no means am I an expert in that. I'm familiar with the topic, but I certainly wouldn't want to, feign, expertise in that. That's a whole, kettle worms that I don't want to touch. Having said that, there is something that I do know about that has a lot to do with this oxygen story that we've been talking about. And that is this phenomenon, this epidemic in the United States of something we call malocclusion. Now, what is malocclusion? Malocclusion is the maldevelopment of the oral cavity and thereby of the airway. Now, the airway is where dentists and doctors meet because they care about the airway. We care about the airway. We both care about the airway. And the bottom line is our babies' airways are not what they used to be. So 2 years ago, I participated in a panel symposium at UCLA called Why the Long Face? And it had to do with a phenomenon which is now known as craniofacial dystrophy. And you've probably seen yourself that a lot of babies and children and young adults have basically elongated faces with short chins. And it's one of the reasons why beards are so common and prevalent in young adults is that they're hiding a short chin. They're hiding their micrognathia. And the question is, what's happened that has led to this? And it turns out that the maternal diet during pregnancy, like fructose, okay, because it's altered energy availability within the growing skeleton, it's actually shortened the chin. Okay, and it's reduced the amount of IGF-1, insulin-like growth factor 1, which would normally elongate the chin. And that's leading to smaller chins, which is leading to this phenomenon of reduction in the size of the craniofacial respiratory complex, the airway.
Now, and so that's not just a cosmetic thing. It's going to affect your ability to breathe and take in oxygen.
Right. It's going to affect oxygen transport through Bernoulli's principle. So think of it this way. You have an ovoid. You have an ellipse. Okay. What shape ellipse conducts the greatest air transport? The answer is a circle, right? A cylinder. Okay. If it's elliptical, it's going to be less because of Bernoulli's principle, So like if it's 5 by 5, that's 25 pi. Whereas if it's, 4 by 6, it's going to be 24 pi instead of 25 pi. So, everything that's not circular is going to mean less cross-sectional area and therefore less fluid, in this case, air transport through that. Okay. So that means that there's less oxygen getting to the lungs, which means there's less oxygen getting to the tissues for burning.
You're more hypoxic, and if it's less burning,
right. And what that means is that number one, it's going to affect your growth, and number two, it's going to increase growth. By the way, but it's going to lower burning. And what that ultimately means is that you are set up for chronic metabolic disease in later life because you can't transport as much oxygen. So what's the telltale sign of that is a high arched palate, okay? Because the tongue is supposed to be pushing the incisive suture at the base of the mouth, at the top of the mouth away in order to create the tongue box for the tongue to live in. And that's going to increase the size of the airway. And so babies, because they're not sucking on mother's breast, but rather on baby bottles or on pouches, are actually not generating the negative pressure to push up against that incisive suture to grow the airway. And that's what's leading to the malocclusion. So not just what babies eat, but how they eat has a lot to do with the risk for chronic metabolic disease in the future. And if you have malocclusion, it's one step, to obstructive sleep apnea.
Yeah. So it's not just about, so what we ingest affects stuff that's happening at the level of biochemistry, the nutrients and things that go into the body. There's also mechanical stimulation that's necessary for the proper physical spatial development of things, which goes on to affect things like how much oxygen you're taking in and so forth. So all of that stuff is important. It's not just a cosmetic thing if it's a physical malformation.
Except those two things are tied through ultra-processed food. So ultra-processed food is causing problems at both ends of the spectrum. What we eat and how we eat. Both.
Right.
It's the combination of the two.
Yeah. And so, another way of saying all this is, you lose on both ends with ultra-processed food.
Yes, you do.
Even if you were to make a baby's bottle that had the perfect physical properties to mimic a nipple, you'd still have the formula in it. Or if we do the reverse of that, even if you had the perfect breast milk, replicated and put into the formula, the physical stimulation would be different. And so you'd lose on one end in that case.
Exactly right. And so we're seeing that throughout, virtually every developed country.
One of the things I want to spend some time talking about in the time we have left is going to be focused mostly on brain stuff. So obviously the brain is a very expensive organ in terms of, its weight compared to how much of the body's energy it uses. It's a very complex and delicate organ. It's very sophisticated. And so part of what that means is there's many ways that it can break.
You're telling me.
When we, what would be a good way to lead into some of this stuff? So the brain is an energy intensive organ. That's something we need to keep in mind.
Lots of mitochondria.
Boatloads.
More than any other organ.
And our neurons, for the most part, don't divide. So when you kill them, you can't replace them like you can with many other cells.
That's right. They're very easily killed.
They're very easily killed. So they're delicate, they're complex, they've got a lot of mitochondria. So this is the potential for a lot of energy problems, a lot of potential for reactive oxygen species. They can't divide, they can't be replaced. I've started to hear more, there's a lot more crosstalk that's happening now between the neuroscience and brain world and the metabolism world that, we used to think of these as two separate domains, but obviously they're not.
They're not.
I've started to hear things about various forms of neurodegeneration. You might be able to think of them as forms of insulin resistance of the brain or metabolic impairment of the brain.
Yeah.
How do we start to think about the brain from a metabolic perspective?
Yeah. I don't like that insulin resistance of the brain thing personally. Metabolic dysfunction of the brain, yes, absolutely. But insulin resistance of the brain, I actually don't like that term. I would prefer not to use it. But yes, no, this is a big problem. Think about it. Your brain has more mitochondria than anywhere else. 2%, your brain is 2% of your body, but it uses 20% of the energy. Okay. The brain is the most energy intensive organ in the body by far. Nothing comes even close. Worse yet, the brain has no place for energy storage. So it has to have a constant supply of energy being brought to it. Now, in general, that's glucose. And if you're on a ketogenic diet, it can be ketones. Your brain can ultimately use both. But, for the most part, it's glucose. And we're not going to go into the ketogenic diet thing right now because we don't have time for that. Okay. But the bottom line is your mitochondria have to burn that glucose to ATP, as we talked about at the beginning of the podcast, right? Now, mitochondria do that. You get 36 ATP out of every molecule of glucose, right? But you also get something else that you didn't want. You have oxygen species, ROSs. Turns out ROSs are not benign. ROSs are like if you're burning a firewood, okay, you get the fire, but you also get the smoke.
Hold on a sec, Robert. The sound cut
And
out
so,
for me.
sorry?
So things just paused. There was a hiccup in
Yeah,
the
I saw that.
technology.
I saw that.
Okay, it's good now. So I think you can go.
All right. Well, I'll back it up so that so mitochondria burn glucose, and glucose is essential, for normal, brain function. But think of it this way: if you're burning a piece of firewood, you're getting the fire,
Right.
but you're also getting the
The
smoke,
toxic smoke. Right. It's
and the
inevitable.
smoke is, part and parcel. It's, you can't undo that. So you got to do something with the smoke.
Mhm.
Now, yeah, that's why we have chimneys,
Right.
right?
Right.
Okay, get rid of the smoke. Well, the fact is, the cell makes smoke, makes something called ROSs, reactive oxygen species. Inside the mitochondria, there are 11 separate steps, and every one of them generates reactive oxygen species. So, yes, mitochondria are an ATP machine. They're also an ROS machine, both.
Yep. You have to
The,
burn. Burning necessarily makes smoke, but the cell is probably equipped in some way to, it's got the cellular equivalent of chimneys and smoke detectors and these things.
well, it has antioxidants. That's what it has. So the ROSs go to a specific organelle in the cell called the peroxisome. And the peroxisome is where the antioxidants are. And that's where they go to die or be quenched, as we refer to it in biochemistry.
So they run into antioxidants in the peroxisome. And if they didn't do that, they'd run into something else in the cell, which they would probably break.
That's right. Exactly right. So the ROSs can cause lipid peroxidation, protein denaturation, ultimately cellular dysfunction, and ultimately cell death. So you got to get rid of the ROSs. You got to get rid of the ROSs. And the standard way you get rid of the ROSs is with antioxidants. And that's why it's important to eat antioxidant rich foods. Okay. That's true. But sometimes the antioxidants don't even get to where they need to go. And sometimes some cells are going to be antioxidant poor, whether you like it or not. And the question is, what do those ROSs do? Now, in high dose, they can be damaging to the cell. They can lead to, fat denaturation, lipid peroxidation, et cetera. But they're also signaling molecules. Remember, we started out by talking about the kinases and the phosphorylation status of these enzymes, right? Remember?
Yep.
Okay. ROSs drive that phosphorylation phenomenon also.
Is it
And
because,
so,
is the way to think about this, for example, if we think about the AMP kinase pathway, that's how much mitochondria I have, how much ATP I'm producing, and therefore the level of ROS you're producing is going to be a kind of readout of that part of the equation.
exactly right. Exactly right. And so when the ROSs go up in the cell, what that's telling the cell is, hey, I got enough coming down this way here. All right. I can't handle the entire, the load.
Yeah, there's too much
All right?
smoke. The fire is
Too
big
much
enough.
smoke. Right. Lay off and divert. Okay. And so you end up sending that glucose, which should have become ATP, down different pathways, including the ones we talked about before, the growth pathways, the pentose phosphate shunt, the Randall cycle, the de novo lipogenesis, or, just plain old storage. In the liver, store as fat. In the brain, stored as glycogen. Okay, your brain's not supposed to have glycogen, but Alzheimer's patients have glycogen, which is kind of like weird. Why is there glycogen there? It's because you couldn't shunt it down the mitochondria because the mitochondria are broken. Okay, that's what we've learned about Alzheimer's is that, mitochondria is a primary mitochondrial problem. All right, so think of it this way. You've got your diet driving ROS formation through the mitochondria, but you also have the environment driving ROS formation through all of these environmental insults, like the obesogens that we talked about. Every obesogen is an ROS generator. Guess what? Everything that's been associated with Alzheimer's is also an ROS generator, including ApoE4. They're all ROS generators. So what's happening is you've got ROSs from within, from the mitochondria, and you've got ROSs from without, from the environment. So you have this ROS pool that's greater than the antioxidant capacity of any individual neuron to be able to detoxify. And what that's going to do is in low dose and acutely, it's going to cause diversion of energy away from mitochondria. So you're not going to generate the ATP you want. And in high dose, ultimately can cause neuronal damage as well.
Yeah. And so even kill the cell.
At low dose, you have mitochondrial dysfunction. At high dose, you have basically cell death. But in either case, that's not good. So you have decreased ATP generation because of the ROS burden on any individual neuron. That's step one. Now let's do step two. Glucocorticoids, sleep deprivation, exogenous glucocorticoids, stress, okay, all generate increased cortisol, increased glucocorticoid burden. So what do glucocorticoids do to neurons? They increase ATP utilization. You take a neuron, you expose it to cortisol, and the need for ATP goes up. This has been known since 1984 when Robert Sapolsky first showed that hippocampal damage occurs in response to high-dose glucocorticoids, but you could stop it by giving extra glucose. And more recently, Martin Picard at Columbia has demonstrated the cerebral hypermetabolism associated with increased steroid exposure. So now what you've got, step one, reduced ATP production because of mitochondrial dysfunction.
And now you've got increased demand.
Step two, you've got increased ATP utilization because of glucocorticoids. Now you have a real energy crisis. Now you don't have enough ATP to do the job. So what would that look like? Brain fog, irritability, depression, cognitive impairment, okay, metabolic syndrome of the brain, as it were. Okay. Now, step three. You have these proteins in each neuron, okay, and in the glia, okay, and this protein is known as amyloid precursor peptide, APP. Now, if you cleave it once, you get two trophic factors that help cells grow and help neurons synapse. If you cleave it three times, you get cells, you get proteins and peptides that actually cause involution. As Dale Bredesen puts it, connection versus protection. And the ATP availability within the cell basically dictates whether that APP gets cleaved once or gets cleaved three times. So the energy availability within the cell is part and parcel of whether you are in connection mode or whether you're in protection mode, whether you're in involution mode. So you are actually losing neurons. You're losing neuronal synapses, sorry, not neuronal synapses, because of the reduction in ATP availability from step one and step two.
Yeah. And it kind of makes sense, right? If the cell's basically like, oh, I don't have enough ATP, certainly you don't want to maintain and grow more connections because those are going to be ATP intensive.
Exactly. That's exactly right. So you're losing connections in that space. And that's going to have its own effects on behavior and also on neurocognition. But that's not all. Now we have step four. And step four is the fact that ROSs directly drive inflammation. They activate NF-kappa B, which is the driver of the inflammatory process. And they're the thing that turn M2 microglia into M1 microglia. And that's the thing that basically turns on the scavenger system to get rid of faulty cells. That's the immune system of the brain, being activated, the inflammatory process within the brain, the neuroinflammation, and it's being driven by the ROSs. So now you have vulnerable neurons with plaques, and you have the inflammatory process turned on, and now you have the clearing of what used to be perfectly good cells, and now you have Alzheimer's.
Is that why, this isn't my field, but I know that there's been a lot of controversy at different levels over the amyloid beta and the plaque story and some of the classic studies that have been done here. But if I'm hearing you right, the plaques are a marker of an underlying energy metabolism problem. And therefore, if you merely were to get rid of the plaques, but not the upstream problem that's originating the mitochondria, you'd still have a problem.
And that's why none of these drugs are working.
Right.
For just that reason. That's why we know that that's the problem. The plaques are the result of the problem. They're not the cause of the problem.
Yeah.
They're the marker that there's a dysfunction. They're not the cause. And until you fix the cause, you can't fix the problem. So we have developed, and full disclosure, we have developed a company around this, an Alzheimer's prevention company called Snap Recall, very specifically to stop this process. And it's personalized, it's individualized, because you have to know where the ROSs are coming from in any individual patient, because there are a myriad number of potential ROS generators that you have to worry about, some of them being diet, some of them being activity, some of them being environmental exposures, the exposome. Okay. And you have to know where they're coming from. And you also have to be able to alter the ATP utilization within any individual. And you also have to be able to, basically turn off that neuroinflammatory response. But you can't turn it off until you know what caused it. So we have developed a paradigm, and we've tested it on 10,000 patients over 10 years to demonstrate that we can actually find the problem 3 years before any cognitive decline. We can actually see synapse-associated phospholipids in the blood 3 years before any change in cognition.
So in other words, you're saying you can measure something in the blood that indicates that the energy problem has already started in the neurons, but has not yet become advanced enough to actually kill the cells.
Right. So we have an early warning system, and we can use AI to actually work backwards based on what those synapse-associated phospholipids are to determine what the pathogenesis, what the pathway was, so we can actually figure out what the cause was in order to be able to prevent it. One thing I am very sure of, Nick, is that Alzheimer's does not have a cure, but it has very, very clear prevention. But the prevention will be different for each patient based on what the driver is.
Yeah. And why do you say that? Is it because by the time someone has Alzheimer's, where it's clearly manifesting in symbols that we can see and identify, they've already lost irreplaceable neural tissue?
Exactly. And you don't rebuild that. Okay. Once you lose it, it's gone. So the only thing you can do is prevent it. So if you know 3 years in advance that you're on that path, you have the opportunity to do something about it.
So, and just to reiterate, so this energy problem that neurons often will eventually face for whatever reason, it will first result in some retraction and some pruning away of synapses and things like this before cell death actually happens. So there is a window of time. And that's right. You guys are looking in this window.
That's exactly right. We're working upstream of the problem before the problem becomes incontrovertible.
So someone gets this blood test, they see they're on this path, or maybe they don't. They just want to prevent the path from ever going down this path.
Yep.
What are the key things, based on your knowledge today, that you would recommend people can do throughout their lives, and especially as they get into middle and late age, to prevent the energy problem and the ROS overload that will eventually lead to Alzheimer's or other forms of neurodegeneration? I'm guessing this issue is a general issue, not an Alzheimer's specific issue.
Oh, yeah, absolutely. And it's for all dementia. It's not just for Alzheimer's, but Alzheimer's is the one everyone knows. I mean, there are 4 dementias. There's frontotemporal, there's Lewy body, there's vascular dementia, and then there's Alzheimer's. Okay. But they all have, are ROS drivers, being driven by ROSs in any case. So this goes for all of them. So the short answer is all the things you already know about that basically improve metabolic syndrome. Diet. Okay. You got to get rid of the ROS generators. The ultra processed food diet, the Western diet, is an ROS generator. Okay. It was built to be so. So that's number one. Number two, sleep. Sleep is essential. Now, sleep is a problem for a lot of people. And the question is, what do you do about it? Different causes need different treatments. Okay. And that is a whole issue in and of itself that we will undertake for all these people, to figure that out. Number three, okay, exercise. Why exercise? Exercise generates ROSs, right? Okay, but what exercise does is it also increases trophic factors within the brain that actually stabilize those synapses, like for instance BDNF. Okay, so that's essential. Okay. And number four, socialization. Okay. You got to get the cortisol down. If you don't get the cortisol down, nothing else is going to help. Okay. So the question is, why is everyone's cortisol so high? Well, because everyone's lonely.
Yeah, we're social primates. That's the kind of animal we are. You need to have a social network.
That's right. And so the question is, what's going on inside your head that's leading to your internal stress and your increased utilization of glucose because of your cortisol problem? That is going to be individual across the board. And, I can't give you a blanket statement for any of those. Okay. But that's why this company exists, is to be able to personalize each person's own therapeutic in order to prevent the process from going any further.
And when you say this company, do you mean there's basically it's like a blood test, or I can order this test and you can tell me, am I down this path? And if I am, what specific path am I on and how do I get off it?
So we used to be just an analyte company that offered the test. We are now an Alzheimer's prevention company in order to be able to basically take patients from start to finish all the way through. So we have an assessment, we have a therapeutics, and we have a monitoring component, all built in to basically keep people from developing neurocognitive dysfunction and neurocognitive decline across the board.
How much does such a test cost?
Only $300. It's actually cheaper than A beta and phospho-tau.
And I want to ask, so I want to go back to this concept of mitochondria burn. When you make fire, there's going to be smoke. There's going to be some amount of reactive oxygen species production. It's inevitable, and it's actually used as a signal by our cells. So it's not all bad. That's used as a signal. Right. No. Our cells take advantage of the fact that it's inevitable. And it's really about the antioxidant capacity, right? When you go past your cell's antioxidant capacity, then you start to do more damage, but you're never going to get your ROS production down to zero. That's not possible. We also said things like stressors like exercise acutely increase ROS production. So what I want to ask you about is basically, how much is too much? How does one assess the appropriate amount of stress? What's the difference between overtraining versus exercising and it's healthy? What's the difference between too much and too little here? How does an ordinary person detect this in their life?
Yeah, that's hard. So I'm actually writing a book with my colleague, Dr. Alyssa Eppel, who's a cognitive psychologist here at UCSF. We're writing a book right now on the amygdala, which is basically the stress generator of the brain. Okay, and it's called Freeing the Hostage Brain. And so you'll invite me back to talk about that book, no doubt in the future when it's released. But there are two kinds of stress. There's adaptive stress and there's chronic toxic stress. They are not the same, and they don't do the same things. And they ultimately impact on different pathways within each cell and specifically within the brain and specifically within the amygdala to ultimately lead to systemic and therefore mental health changes. And different people are going to have different thresholds where those things basically break down. Example of that is pilots. Okay, pilots are the ultimate multitaskers. They can multitask, their way out of everything, right? Well, not really. Turns out you can ultimately overload a pilot just like you can overload, your grandmother. Okay. You can basically give them enough stuff to do all at the same time that they will end up in this same chronic toxic stress mode and have the same feelings of fatigue and burnout as everybody else. They just have it at a much higher level. Now, why they're able to process all that stress when, your grandmother can't, that we still don't know. But ultimately, everybody has their breaking point. And we don't know what causes that.
I want to spend just a few minutes talking about the topic of the ingestion of obesogens or other things that increase the ROS burden of the body. Some of these might be obesogens that have metabolic effects that we can talk about. Those might be chemical obesogens. Those might be caloric obesogens. There's also things like various drugs or chemicals that people might get exposed to that increase ROS production, but may or may not be obesogens. Sure. One of the hooks I want to use here, because it's been in the news recently and a lot of people are exposed to this on purpose all the time. So that's the general topic we'll get to. We can take it wherever you want, but I'm going to start out by asking you the Tylenol question here. What's going on with Tylenol? And is there something to the story that this could be affecting development in ways that might influence autism and or any number of other issues that arise in development?
Right. So one of my favorite topics. So RFK said Tylenol is the cause of autism. And then Trump doubled down and said, don't even take Tylenol if you're pregnant. What the hell's going on? What the hell's going on? All right, here's what's going on. Tylenol causes ROSs, big time. In fact, we pediatricians knew this before anybody else because we would see patients, 3-year-olds, 4-year-olds show up in the pediatric emergency room with Tylenol toxicity because, the kid got to, the parents' Tylenol stash in the, in the medicine cabinet. Okay. And it would fry their liver and they die from, hepatic necrosis from the ROS buildup from acetaminophen. Okay. And the only thing that you could do to stop it was to give a very potent antioxidant called N-acetylcysteine, called Mucomyst. And the reason is because cysteine has a sulfhydryl group, and so it is a very good ROS scavenger. It's one of the best. Okay, and so that's why Tylenol toxicity needed Mucomyst. So we've known about this like forever. Well, the question is, could Tylenol ingestion during pregnancy lead to increased ROSs, and could increased ROSs lead to cell damage in the fetus, which could ultimately express as autism? That's the question. And the answer to that is sort of, a little. Okay, like everything RFK says, there's a nugget of truth in there, and then everything else is overblown, and he actually loses the thread. And here's why. If you look at the data, okay, of the meta-analyses that have been done on Tylenol and autism, the hazard risk ratio for Tylenol ingestion by the mother and autism in the fetus, in the offspring, is somewhere between 1.05 and 1.2. Now, public health officials agreed years and years and years ago that you would need to reach a hazard risk ratio of 1.3, so a 30% increase in risk, in order for it to be a public health issue.
Yep. So it needs to be there, but it needs to be above a certain threshold before it becomes a public health issue. I guess from a policy standpoint, the idea that we need the problem to be big enough to divert national level resources to it because
Right.
there's so many tiny problems. There's only
That's
so
right.
much we can do.
Exactly. So 1.05 to 1.2 doesn't reach that threshold. But nonetheless,
It's
the
there.
signal's there. The signal is there. There is a signal. Okay. So it's not like, RFK divined this out of his head. The signal is there. The question is it enough to do anything about? So now that we understand that it's this ROS problem, well, there are a whole bunch of things that cause ROS generation. Glyphosate
Uh-oh.
radiation causes ROS pollution. And, every one of these insecticides that, are in our food cause ROS generation. Tributyl tin causes ROS generation. It's
Right.
in the water. Okay.
Yeah.
If you
It's not
took,
that Tylenol doesn't do this. It's that a
it's like
bajillion
everything
things
else
do it.
does
And
more.
yeah, yeah.
Okay. So taking Tylenol out of the armamentarium is not going to solve the ROS problem. And if you don't solve the ROS problem, you're not going to solve the autism problem. And Tylenol is such, it's a drop in the bucket compared to everything else that's going on. And also, what are you going to do for mothers who have a high fever, during
Hmm.
pregnancy?
Hmm.
What are they going to be treated with?
Yeah,
That is an ROS generator by itself
yeah.
and
So
could do its own damage.
yeah, so
You know,
lots of our ROS burden's high, and there's many reasons for it. There's certainly not just one drug that's causing this. There's many
that's right.
things in the environment.
And
Totally agree.
how do we know that this is a driver of autism? Okay.
Okay.
The answer is if you look at the scientific literature, the single best model for human autism is the exposure of rodents and other species as well to valproate, to valproic acid or Depakote, which is an anti-seizure medicine. We use it in children even today. Okay. But if you want to generate autism in an animal model, you give them Depakote, and the hazard risk ratio goes up enormous. And the reason is because Depakote is a driver of ROSs like nobody else. So we know that increased ROS in utero means risk for autism. And yes, Tylenol is a modulator of that. I'm not saying it's not, but it is paltry in comparison to all of the other ROSs that the pregnant mother and the fetus are exposed to. That basically telling people to stop taking Tylenol because of a risk for autism is, unconscionable and really, as far as I'm concerned, immoral.
Mhm. One thing I want to ask you about. So this will be specific to what we just talked about, but there's also just a more general thing here that I want to get people thinking about. So you said that the hazard ratio for Tylenol was 1.05 to 1.2. So not nothing. It's significant.
Not
It's
nothing.
there, but
It's there.
it's not huge. It's not above this 1.3
Right.
threshold that people like to point to as something being, a very high hazard ratio. So it
Right.
is there.
Right.
Now, the question is it that hazard ratio for everyone? Because the average person actually will tend to conform to the average number computed in these analyses. Or is it that you've got multiple subpopulations of people, and for some of them, this is a huge ROS generator, and for others, it's not, and we're just sort of averaging those away so that the overall is kind of low?
Maybe. We don't know. Is it possible that the antioxidant capacity of those fetal neurons is ultimately, what's at play here? And that, if you had an increased antioxidant capacity in those fetuses, they could stave off, a Tylenol insult and not have a problem. And maybe mother's diet could contribute to that because that would increase their, antioxidant capacity. We don't know. There's no data on that. We could do research on that. That's possible. We could answer that question, maybe. It'd be hard, but not impossible. But right now, we don't know the answer to that. I can't tell you why one kid might be at risk for Tylenol autism and another kid might not be at risk for Tylenol autism. I don't know the answer
Mm-hmm.
to that.
Mm-hmm.
Okay, that's above my pay grade.
So, given that the modern environment has so many different things in it that are essentially poisoning our cells and our mitochondria,
Yeah.
given that there's so many things that will increase the ROS, the reactive oxygen species burden of our cells, which is going to cause all kinds of, this is a very general thing. So it's going to cause all kinds of problems and predispose us to all kinds of issues at any stage of life. What can we tell people in terms of how they can generally reduce their ROS burden if it's, in everything, in the air, in the water, in the food? What are the basic simple things that people can do that are going to best and most easily buffer them against this pervasive problem?
Yeah. So a lot of it's baked into the cake. We can't do anything about ionizing radiation. We can't do anything about microplastics. We can't do anything about air pollution, short of what we've already done. Okay. A lot of those things, we can't do anything about. What could we do something about? Well, we could do something about our food. That we could do. And of all the ROS generators in our food, sugar is the biggest one. And the reason is because every time a fructose molecule binds to a protein, you get an ROS. And so the more fructose, the more binding, the more ROSs. And that's been shown 50 ways from Sunday. And that causes metabolic damage. So in the same way that reducing sugar will reduce metabolic disease, reducing sugar will also reduce ROSs in part because those two things are related, and that includes in the brain as well. So to me, as I said earlier, of all of the different manipulations, of all of the different public health, responses that we could take to this metabolic syndrome epidemic, to this dementia epidemic, to this ROS generation epidemic, getting the sugar out of the food is job one. Mm-hmm. Because it's the 2,000-pound gorilla in the story. And everything else is, poultry compared to that. I basically put sugar at about 80% of the problem. So there's still 20% of the problem, but, it's only 20%. The rest of it is, it could be fixed by fixing the sugar, issue in food today.
Yeah. One thing I want to ask you about, Robert, and I should have done this earlier, it just slipped my mind. Might take a few minutes. Everything that we've said about fructose and ROS production and the metabolic issues that can be caused by having too much fructose, people could argue and nitpick about how much is too much in all this. Generally in biology, right? I like to think in terms of the story of Goldilocks, especially when I'm explaining things to people. Not too much, not too little. Usually there's a happy medium of things. Right. You can certainly overdo fructose. Is it possible to underdo fructose? No. What is fructose good for? Could someone completely eliminate fructose from their diet and have no downside? That's one way of asking it.
Absolutely. So there is a disorder called hereditary fructose intolerance, HFI. Okay. So this is an enzyme disorder of a enzyme in the liver called aldolase B. And this is one of the enzymes necessary to metabolize fructose down to pyruvate. And so when you are missing this enzyme, you can't convert the fructose all the way to pyruvate. And what happens is you end up hypoglycemic. So these are babies who are born normal, look fine, everything's good. They're eating breast milk, or for that matter, formula, doesn't matter. Okay. And then they get their first taste of juice at 4 months to 6 months, somewhere in there. And all of a sudden, they have a seizure at home. And the parents go ballistic and they go crazy and they call 911 and the paramedics come and they check a glucose, by glucometer, and the kid's got a blood glucose of 5, when it should be 70. And so they give the kid glucose by IV and they show up in the emergency room. And then the doctor is trying to figure out what the hell happened to this baby, that the blood glucose was 5 and that the seizure occurred. Okay. And depending on how long the seizure occurred, depends on how many neurons the kid lost.
Hmm.
Okay. Because these kids invariably end up severely retarded because of the insult that they took, which is a disaster. Okay. And some of them die because of it. Now, they do a liver biopsy and they assess all the enzymes and they find out they're missing aldolase B. And so now they know that they have hereditary fructose intolerance. That means that there's a very good chance that any other babies born to that parent, to that set of parents, are going to have it also. So you put these babies that haven't been born yet on a fructose-free diet from day one, from day one. And guess what? Not only are they protected from this horrible disease, but they end up actually smarter than everybody else.
Hold on. Before we get to that, I want to make sure I'm understanding the actual issue here, the nuts and bolts. So you've got a mutation, a problem in an enzyme that metabolizes fructose. So you can't get rid of the fructose if it comes in, but this is causing them to be hypoglycemic and they need glucose. What's the actual connection there? Why are they hypoglycemic if they can't get rid of the fructose?
That's a complicated story and has a lot to do with the biochemistry of the glycolytic pathway.
Okay, but it works out that way.
It just worked. I mean, I could do it, but I'll floor everybody in the room.
If you can't process fructose, you will end up becoming hypoglycemic.
Yes.
And if you take a lot of fructose, like these children sometimes do, because they don't realize they have the disorder yet, bad things happen. Seizures and
an example of this in not in babies, but in humans, in adults, is Jamaican ackee fruit. So I don't know how much you know about Jamaican ackee fruit, Nick. Jamaican ackee fruit is the national dish in Jamaica. Okay. But the ackee tree, okay, the fruit has to mature. And when it matures, it opens up and the fruit becomes obvious and then falls to the ground. And you basically pick up the fruit from the ground and you turn it into the Jamaican national dish. You can buy Jamaican ackee fruit in Jamaican groceries around the world. Okay? But if you eat an immature ackee fruit, there is a compound in the immature fruit that has to be metabolized away. That compound is called hypoglycin. And basically it acts on aldolase B and causes these people to end up having their blood glucose goes from 70 to 5, just like the babies did. Okay. And we have a name for this. It's called Jamaican vomiting sickness. This is Jamaican vomiting sickness from eating immature ackee fruit, which basically poisons that same enzyme. And you end up with the same hypoglycemia, and you can die from that. And so everybody in Jamaica knows that, of course.
Yeah. So if you mess with the ability of the body to metabolize fructose, it can lead to hypoglycemia, and your cells are all running on your sugar. And so if you take your blood glucose suddenly, then you've got a problem.
That's right.
A big problem.
So these babies who, the probands of this union, they don't know that until it happens. Okay. But then that, not to let that happen to any babies born after that. And so they all end up just fine on a pure fructose-free diet from day one. Now, how do I know all this?
Yeah.
I am the chief science officer and co-founder of a nonprofit here in the Bay Area called Eat Real. And our job is to get real food into K to 12 around the country. And we're doing a pretty darn good job of it. And I'm very proud of the work we do. Years ago, we had a board member who, in fact, had hereditary fructose intolerance. Her sister was institutionalized, and the parents knew that this, my friend Nadia, that she might have the same thing. And so she had a liver biopsy when she was a baby. And indeed, she has the same disorder. And so she was basically put on a fructose-free diet. She's never had a lick of an ice cream cone, or a lollipop in her entire life. And Nadia became a PhD molecular biologist and also worked the M&A desk at Goldman Sachs in England before moving to the United States to start her own company on Wall Street. Okay. And so she told me her whole story. And I was like, wow. She says, that's why I'm so interested in nutrition. I said, yeah, I get it. So, in fact, there's a registry of HFI patients that's kept at UC Berkeley. And so we have the patients who were the probands who were all basically decimated. And we have the patients who were the, products of, later unions where they knew the problem. And so those patients are being followed and they're all doing just fine. So you do not need any fructose. Anybody who says you do is full of shit. Okay, you do not. Just like you do not need any alcohol. And anyone who says you do is also full of shit. Now, our livers have an innate capacity to be able to metabolize some, but when you overdo it, you get into problems. Same thing with fructose.
Right now, I would imagine that's because natural sugar is often in the form of sucrose. So you're going to eat some fructose if you're out in the wild living. So the body must have evolved some capacity to deal with it, but that doesn't mean it's needed for anything critical.
And also the fruit has the fiber, and the fiber actually limits the fructose absorption from the gut into the bloodstream in the first place. So really, when you're consuming fructose in fruit, you're really basically providing that fructose to your bacteria, not to your liver.
Yeah, because it's not being absorbed as readily because of the fiber, more of it is likely to be metabolized in the gut by other bugs before it actually gets into your bloodstream.
Exactly right. So I'm not worried about the sugar in fruit. I'm worried about the sugar that's been put in the food by the food industry. That's what I'm trying to fix.
And you mentioned briefly that the kids with that disorder of fructose metabolism, if they're on a fructose-free diet from day one, they end up smarter?
Yes, they end up smarter. Right.
Like on average than a normal person?
Indeed.
What does that mean precisely?
It means they're smart. I mean, that means they have higher IQs.
Oh, wow.
Yeah. So it may be that the fructose that babies are normally exposed to is actually, making them dumber.
Wow.
We don't know that for sure. I mean, we would need a study to answer that, but I mean,
that population, that's suggestive. That population does have a higher IQ when you measure it.
Uh-huh.
Wow.
Yeah, kind of, like knock your socks off.
Well, Bob, I want to thank you for your time and for coming back onto the show. Any final thoughts you want to leave people with, or anything you want to reiterate from our discussion today about obesogens or ROS production?
Basically, what I want people to understand is that the biochemistry matters. Now, I know, Nick, you know that. And that's why you do this podcast. And that's why I'm happy to appear on it. Okay. But people really need to understand that the biochemistry matters. And when you understand it, you can actually figure out what's going on. Right now, we are in this period of mistrust, of misinformation, of, basically degrading science. And it is doing nobody any good. And ultimately, I think it's going to do mega harm. We need to be teaching science, across the board. And we need to be teaching civics across the board so that people understand what it is like to actually share society with other people. Right now, we're in a very bad place. And I think it's because our education system in America has broken down and is not doing what it's supposed to do. The science matters. The biochemistry matters. And when you understand these things, you can actually not just help yourself, but help your family, your community, and ultimately all of society. That's my job.
All right. Dr. Robert Lustig, thank you once again for your time.
Thank you for having me.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.